Thanks, Federica, for a kind introduction. It is my great honor and pleasure to be here. So I'd like to thank the organizers of this 19th ICI meeting for inviting me here to speak. So today I'd like to talk about how Treg can be used for induction of tolerance, self-tolerance, especially to treat autoimmune disease. So it is now well established in these 30 years that the regulatory T cells, the Treg, they are indispensable for the maintenance of immunological self-tolerance and homeostasis. So Treg, they are expressing the FOXP3, transcription factor FOXP3 in the nucleus, and CTLA4 and CD25 are receptor on the cell surface. The majority of them are produced by the thymus, and some of them can differentiate in the periphery, especially in the intestinal mucosa from naive T cells. And they can also be induced in vitro, called iTreg, in the presence of TGF-beta, IL-2, and then antigenic stimulation. So regarding the immune tolerance, it was shown that immunological tolerance is adaptive, acquired, and then antigen-specific, as shown by McFarland Barnett and then Peter Medavar and others. Now, in clinical setting, how we can induce or establish self-tolerance in clinical settings, that is a kind of a long outstanding issue in immunology. So modern immunology has not yet achieved the clinical immunological tolerance. So Treg, they are T cells expressing T cell receptor, so they can recognize antigen. So the question is, how antigen-specific immune tolerance can be re-established with Treg to treat autoimmune and inflammatory diseases? So there are two ways of inducing tolerance by Treg. One is to use a natural Treg. So to expand them in vivo by, for example, low-dose IL-2, because Treg, they are expressing the constitutively and highly expressing high-affinity IL-2 receptor. So low-dose IL-2 can expand them without expanding effector T cells. Another way is to take them out and ex vivo and expand them by antigenic stimulation plus IL-2 and then transfer back to the host. So it can be done with strong antigenic stimulation, such as allogeneic stimulation, but it is difficult with self-antigen. So the other way of inducing tolerance is the convert antigen-specific effector conventional T cells into regulatory T cells. For example, assuming that the actual culprits of an autoimmune disease, they are antigen-primed memory effector T cells. Then, if we can convert antigen-specific effector memory T cells into Treg, and then transfer back to the host, it can be one effective way of treating autoimmune disease and induce tolerance. And so today, I'd like to the basic and clinical aspects of how we can convert the antigen-specific conventional T cells, especially effector memory T cells, into Treg, and then how we can establish or treat autoimmune disease by such the induced Treg. So outline of my talk, let me first talk about the molecular basis of the development of natural Tregs, including thymus-derived Treg and peripherally derived Treg. And then based on this knowledge, let me talk about how the antigen-specific effector memory T cells can be converted to N-Treg-like function, a Treg with N-Treg-like function and functional stability. And then third, with these the induced Treg, how we can treat autoimmune disease in an antigen or disease-specific fashion. So natural Treg development in the thymus, it required two events. One is transcription factor network, including the FOXP3 as a key transcription factor. The other one is epigenetic configuration, including DNA methylation, histomodification, and chromatin remodeling. So at the particular Treg signature genes, a transcription factor bind to that site, but also epigenetic changes at the binding site. Both are required. If we understand these events, and then we can introduce them into antigen-specific conventional T cells, we can perhaps convert them into antigen-specific, functionally stable Treg. So if you look at the natural Treg, thymic Treg development, the immature CD4 signal positive cells, they give rise to pre-Treg. So pre-Treg can be defined as CD25 positive, but FOXP3 negative. And then these pre-Treg differentiate into thymic Treg and T-Treg. They are FOXP3 positive and CD25 positive. And then they express FOXP3. And if you look at the epigenome, what is interesting is at the FOXP3 locus, for example, so this is a very strong enhancer region called Treg-specific super enhancer. And then if you look at the H3K27 acetylation, which is a marker for active enhancer, then very interestingly, it starts from very early on, from double positive stage, and then it becomes stronger along the Treg differentiation. And similarly, H3K4 monomethylation, which is a marker for poised enhancer, again, it starts from double positive stage and becomes stronger along Treg differentiation. So pattern is becoming a bit changed because of an active enhancer modification, or for example, looping. That kind of the changes happens at the specific Treg-specific loci. And the ATAC sequencing, which is, you can look at the chromatin remodeling, and again, it starts from double positive stage, and then it becomes stronger along Treg differentiation. So before FOXP3 expression, somehow Treg are committed to the T cells, developing T cells. They are committed to the Treg lineage. So in addition to histomodification or in the chromatin remodeling, if you look at the DNA hypomethylation or methylation, it is very interesting because if you look at the whole genome for Treg-specific demethylation or methylation, only 300 regions, they are specific for regulatory T cells. And more precisely, if you look at such a different region by bisulfite sequencing, so each strip and each CpG, so this site, they methylated in conventional T cell, but demethylated in natural Treg. And then this region is the FOXP3 CNS2 region at the intron 1. So among 300 regions, you can choose, for example, 5 regions, FOXP3, GITR, CTLA4, AOS, CD25. They are all Treg signature genes. They are all demethylated in natural Treg. And importantly, if you activate conventional T cells, you never see Treg-type epigenetic changes, but in Treg, they very robustly maintained this type of the methylation is maintained. And then induced Treg by TGF-beta, you cannot induce a Treg-type demethylation or retinoic acid or FOXP3 retroviral induction. You don't see the changes. Or memory T cells or IL-2 expanded conventional T cells or Th1, Th2, Th17. So the Treg-specific demethylation, it is highly specific for natural Treg. And then it is FOXP3 independent because here natural Treg, they are expressing FOXP3, and then also you can put in the GFP gene as the FOXP3 locus. And then so natural Treg, they are GFP positive and the FOXP3 protein positive. But here if you put it, the FOXP3 mutant mice, no FOXP3 protein, but you can see GFP positive cells. So these cells are called FOXP3 null Treg, no FOXP3 protein expression. And then you can make, so suffer from a very severe autoimmune disease because of FOXP3 mutation. And so you can make a bone marrow chimera to prevent autoimmunity. And then you can take them out, FOXP3 narrow Treg. And if you look at the demethylation, they do show the Treg type demethylation in the thymus, and it becomes stronger in the periphery. And importantly, if you look at the protein expression, of course, they don't express FOXP3, but on Treg signature genes, protein, they are all expressed without FOXP3, meaning that the Treg signature genes can be activated without FOXP3. So these findings together, developing T cells, they recognize self-antigen expressed in the thymus and become positively selected, and then express the Treg signature genes. First, Treg super enhancer activated, which induce FOXP3, CTLA4, and other Treg signature genes. And at the same time, almost independently, Treg type epigenetic changes. It also happens. And these two events together, you can now you can induce a functional natural Treg. They can develop. So another question is, based on this knowledge, how we can induce this type of the Treg specific FOXP3 expression together with Treg type epigenome into conventional T cells, and then how we can convert them into Treg. So the idea is the FOXP3 expression somehow, and also independently or separately induce Treg type epigenome into antigen-specific conventional T cells. So let me first talk about FOXP3 induction. So recently, we did a genome-wide CRISPR screening for identifying FOXP3 regulators in human iTreg generation. And so 20,000 genes, almost all genes, you can make a guide RNA library and then lentiviral transduction, and then randomly knock out each gene, and then introduce a Cas9 protein, and then so these cells now they are induced to become Treg in the presence of the TGF-beta, IL-2, and the anti-CD3 stimulation. And then you can pick up FOXP3 high population and low population. And then, then you can identify the genes which can upregulate FOXP3 expression or downregulate FOXP3 expression. So here you see HDAC3, RPBJ, IL-4, all when they are knocked out, they upregulate FOXP3 expression. So indeed, RPBJ, it is well known that it is involved in Notch signaling pathway, but a function of the RPBJ in this case is Notch independent. So if you make a FOXP3 knockout, the RPBJ knockout, then you see FOXP3 is upregulated. And then FOXP3 expression is more stabilized, protein is highly expressed, and then Treg suppressive activity can be enhanced. So actually, RPBJ, HDAC3, and then NCOR, they form a repressor complex, and then directly bind to the FOXP3 promoter, and then downregulate the FOXP3 expression. And then we also conducted a chemical screening. So we looked at the screened 5,000 chemical compounds, mainly the tyrosine kinase inhibitors and serine threonine kinase inhibitors, and then put it in the culture, 3-day culture, and whether FOXP3 can be enhanced, upregulated or induced by a particular compound. So this compound, AS2H3619, abbreviated as the AS compound. This one is very interesting because naive T cells, the TGF-beta can induce FOXP3 positive cells. And then without TGF-beta, by neutralizing TGF-beta, this compound, AS compound, can induce FOXP3 positive cells. And more importantly, effector memory population, CD44 high, CD62L, effector memory population, TGF-beta cannot induce FOXP3 in effector memory population, but AS compound can. And then TGF-beta in AS compound. Now, effector memory T cells, 40%, now they can express FOXP3. So here, TH1 condition or TS7 condition, if you put an AS compound, the interferon gamma positive cells, they are reduced, and then FOXP3 positive cells, they become increased. Similarly, under TH17 condition, IL-17 secreting cell reduced, and FOXP3 positive cells enhanced. And similarly, TH2 or TH9 condition, AS compound can induce FOXP3 under this kind of, in the presence of the inflammatory cytokines. So what's the mechanism? So activated T cells, there is a mechanism so to inhibit FOXP3 expression. So TCR signal activates CDK8-19. CDK8-19 is a regulatory module of the mediator complex. So they reversibly bind to the mediator complex and then regulate transcription positively and negatively. So in activated T cells, it inhibits STAT5 expression, so inhibits FOXP3 expression. But if you inhibit CDK8-19, this is the ACE compound, and then STAT5 become activated, and then induce FOXP3. So here, just show in the presence of AS compound, and with antigen stimulation, so this is obvious specific and TCR transgenic T cells. So without AS compound, nothing happened. But in the presence of AS compound, plus antigen stimulation, those T cells which are expressing the transgenic TCR, they specifically express FOXP3. And if you stimulate four T cells, whether expressing the transgenic TCR or not, they can express FOXP3. So CDK8-9 inhibitor or RPBJ inhibitor or TGF-beta, these compounds can induce FOXP3, especially in effector memory T cells. How about the Treg type epigenetic changes? So what we did is this very simple experiment. So this is a plate-bound anti-CD3, and then in the presence of IL-2 and TGF-beta, plus the agonistic anti-CD28 antibody as a co-stimulatory signal. So here, with co-stimulation, CD28 co-stimulation, and then IL-2 and then TGF-beta, you can induce conventional iTreg. But here, without co-stimulation, now the FOXP3 expression is better, higher, and then they are resistant to apoptosis, and then they can expand, and then more importantly, without co-stimulation, you can induce Treg type epigenetic changes, as shown here. So like natural Treg. Just a conventional iTreg is induced by TGF-beta alone, you cannot see such a Treg type demethylation. So what's the mechanism? So here, the CD28 signal activates PKC and then NF-kappa B. And so if you activate PKC with PMA, then you can inhibit the demethylation. And similarly, TNF receptor signal, for example, CD70 or TNF-alpha, can inhibit NF-kappa B. So if you look at the FOXP3 CNS gene locus, and if you look by CRISPR-Cas9 technology, if you remove NF-kappa B binding site, you can induce Treg type genetic changes. Indeed, FOXP3 minus Treg, the expression of the TNF-alpha NF-kappa B signal dependent gene expression is become lower. And then if you look at the C-rel expression, CD28 minus iTreg, they are lower in C-rel expression like a natural Treg. So, and then the effector memory population. Now we can induce the Treg type genetic changes in effector memory population by removing a CD28 signal, CD28 minus. So you can you can induce FOXP3 positive cells as shown here, and they show Treg type demethylation. And then these cells, when they are transferred in vivo, they are stable. So with CD28 plus a conventional Treg, and then CD28 minus Treg, iTreg, when they are transferred into normal mice, and then 2 weeks later, you can see they are present 2 weeks later. But if you look at the FOXP3 expression, conventional Treg, they lose, they have lost the FOXP3 expression. So 60% of cells, they are now FOXP3 negative. In contrast, CD28 minus iTreg, still they are constitutively and stably expressing FOXP3, and then Treg type demethylation, and then also they can expand in vivo. So now we can induce FOXP3 and then also install Treg type epigenetic changes into the antigen-specific conventional T cells. So the first stimulation without co-stimulation in the presence of IL-2 and then also CDK8/19 inhibitor, that is Senexin A, and then you can induce only 100% the regulatory T cells. We call them SF-iTreg, stable and functional iTreg. They can expand in vivo. This red line, very robustly, you can expand them in vitro. Now, the effector T cells, whether any effector T cells can be converted to Treg. Here, this is an DNFB skin sensitization for contact hypersensitivity. And then if you stain for CD44 and CXCR3, and then Th1 cells, they are expressing CXCR3, and then they are expressing Tbet. And then from naive T cells, it's easy to induce FOXP3 positive cells, almost 100% with Treg type epigenetic changes. From Th1 cells, you can induce FOXP3 positive cells, but still some of them are producing interferon gamma. And then they show Treg type epigenetic changes and are still expressing Tbet. And then suppressive activity is less potent, perhaps because interferon gamma interfere with FOXP3 activity. So, but if you one more cycle, if you convert them one more cycle, and then they stop producing interferon gamma, and then you can make the Th1 type regulatory T cells. How about the T17 cells? This is a mock immunization for inducing EAE, experimental allergic encephalomyelitis. And then again, Th17 cells, this is IL-17 GFP positive cells. They are expressing ROR gamma T, and then from naive T cells, it's easy to induce FOXP3 positive cells, almost 100%. From Th17 cells, we can induce FOXP3 positive cells, but still some of them are producing IL-17. But then they are more suppressive than a naive derived Treg, and expressing ROR gamma T. So Tbet and ROR gamma T expression, perhaps they are good for their migration to the particular Th1 or Th17 type inflammation site. So with these findings, now how we can convert the disease-specific T cells into Treg. So we are working on Pemphigus vulgaris. This is a skin autoimmune disease. A target antigen is the desmoglein 3, DSG3. This is the only one target in Pemphigus vulgaris. So it is good for tracing or monitoring the antigen-specific Treg activity. So in PV, the IgG autoantibody disrupt the keratinocyte adhesion and then form this kind of blisters. In collaboration with the people in dermatology department in Keio University, we worked on the animal model of PV. So they established a very nice system for the self-tolerance. So this is desmoglein 3 in knockout mice, and if you immunize knockout mice with recombinant desmoglein 3, now T and B cells become activated, and then you transfer them into lung minus mice, And then lung mice, of course, they are expressing the desmoglein 3. And then you'll see autoantibody formation and blister formation. So the idea is when we prepare a desmoglein 3 specific transgenic T cells, and then we convert them into stable and functional iTreg, and then transfer to these PVE model mouse, what would happen? So here, when the PVE pathology starts to develop day 7, and then if you inoculate an SF-iTreg, now you can prevent the autoantibody formation and then also clinical development of PV, as shown here, the autoantibody deposition or the blister formation in controlled mice can be suppressed. And then these SF-iTreg, desmoglein 3 specific ones, they can migrate to skin lymph node, but not to mesenteric lymph node. And then at the skin lymph node, they become activated, so they are highly expressing CTLA4, LANG3, and some of them producing IL-10. And then in vitro, if you look at the desmoglein 3 specific autoantibody formation by ELISpot assay, so control mice, you see lots of B cells producing autoantibodies. But here, SF-iTreg inoculated mice, you see the reduction, suppression of an autoantibody formation. But the total B cells not changed. Even the SF-iTreg inoculated mice, they show the higher number of the B cells. And then if you look at the follicular helper T cells, so CXCR5 positive and the PD-1 positive cells, they are for TFH cells. And then with SF-iTreg inoculation, you can suppress the autoantibody TFH expansion. How about GVHD? So we transferred the bone marrow plus allogeneic T cells together with SF-iTreg into irradiated mice. And then now these SF-iTreg, the blue one and then green one, they can suppress, the blue one can suppress GVHD development, like similar manner as natural Treg. So what is interesting is that naive T cell derived SF-iTreg, they are stronger in suppression compared with effector memory derived SF-iTreg. And this is because allogenic the reactivity that is higher in that naive Treg compared with effector memory population. So effector memory population already they have differentiated. So less they show less alloreactivity. So again, if the original conventional T cells are, if they are highly reactive with self-antigen or other antigen, and when they are converted to Treg, they are stronger in suppression. So this is IBD. So one advantage of this SF-iTreg, we can induce IL-10 production by a chemical compound. For example, dexamethasone or cyclic AMP analog. Now you can induce IL-10 producing cells. They are very much effective in suppressing colitis, IBD. So here is pathology. These IL-10 producing Treg, they are very effective in suppressing colitis. Now, based on these mouse studies, we can now induce the SF-iTreg from human donor cells, so peripheral blood. With a similar protocol as in mice, we can induce SF-iTreg. They are highly expressing FOXP3 like a natural Treg, higher than natural Treg. They show Treg type genetic changes, as shown here, 8-ax sequencing, natural Treg and SF-iTreg, both can similar the chromatin remodeling. And then transcriptome, FOXP3, the CD25, they are higher than natural Treg. They are expressing the chemical receptors, so they are prone to migrate to the inflammation site. They don't produce an IL-2 or interferon gamma inflammatory cytokines. So from a PV patient, you can induce SF-iTreg like healthy donors. So they are suppressive in vitro, and then here 11 patients with pemphigus vulgaris, all of them, when they are converted to SFI Treg, they show good suppressive activity. Now, so PV is a kind of exceptional because target antigen is only one. But in many autoimmune diseases, target antigens are multiple. For example, type 1 diabetes, more than 4, including insulin or glutamic acid decarboxylase and so on and so forth. And then SLE, more than 10. So, and then the target antigen, main target antigen may be different among the patients. Then the advantage of SFI-Treg conversion is that we can convert 4 effector memory population into Treg. And then after transfer into the host, those stimulated by antigen, they can expand. But then without stimulation, they'll die. So eventually, we can induce or establish antigen-specific tolerance. And then also, for example, Crohn's disease. It is well known that T cells are involved, but then we don't know yet whether Crohn's disease is the autoimmune disease or not. But still, if T cells are involved, we can convert them to SFI Treg, and then they can be used for treatment. To sum up my talk, I have talked about the effector memory T cells mediating disease. They are antigen-specific or disease-specific, and they can be converted to functionally stable Treg in terms of hospice induction and also the induction of Treg type epigenetic changes. So these Treg can be used for as adoptive cell therapy of various immunological diseases, especially autoimmune diseases, and also in a transplantation setting to prevent graft rejection or severe allergy, to prevent the allergy development. So these findings, let me acknowledge my colleagues, my students, Ryoji Kawakami and then Norihisa Mikami. They are main player of the Treg conversion to SFI Treg. And then epigenetic the analysis of Treg, it was conducted by Naganari Okura and the Yoko Kitagawa. And the recent the CRISPR screening of the genome that was conducted by Kelvin Chen. And then also it is for AS compound, for development of AS compound. This is collaboration with Shu Narumiya's group in Kyoto University, and then the pemphigus vulgaris study. That is the collaboration with people in Keio University, especially the PhD course student Miho Mukai. Thank you very much for your attention.